Wonderful. Thank you so much for having us here. So, Kevin and I are delighted to introduce you to the Monarch Knowledge Craft and Services, one of the Monarch foundational projects. And today we'll take you a tour on the work that is happening across the realm by a wonderful group of people. So, Kevin, next slide, please. Uh, and so we'll start with this. In the beginning, there was chaos. Before the Monarch Initiative started working together, we had these, collections of disconnected databases. Many of these probably result very familiar to you. Next slide, please. And not just databases, but also there started to grow an incredible mass of knowledge about the information in these databases. And so ontologies started to surge here and there in order to be able to bring this information together, applying that semantic layer to the information that we were collecting. Next slide, please. And so what the Monarch Initiative has tried to do is put together this vision of an integrated knowledge graph. On the left, we kind of describe what these things are, right? Like integrating everything in one place, having a unified data model, having unified ontologies and connected knowledge. But before I tell you exactly how we're doing this, I'd like to share just a very quick background about what a knowledge graph is. Next, please. So this is a way of representing information as a network of interconnected nodes and edges. And the nodes can represent entities like genes and diseases or drugs. And the edges represent the relationships that happen between these entities. And so, for instance, an edge might connect a gene with a disease that it causes. And in the case of the knowledge graph for Monarch, it could help us identify new connections between diseases and their potential causes. And this can be, I think, especially helpful for investigations on rare diseases where it is very often there's very little data available. So ideally, knowledge graphs will also help researchers in general to identify potential treatments for diseases. And by looking for the connections between a rare disease and other better understood diseases, maybe we are able to identify existing treatments that could be repurposed for certain rare diseases. Click, please. And so how it happens is that we integrate data into a knowledge graph, and this offers significant advantages over traditional relational databases. So for instance, the knowledge graph has a schema-less or flexible schema design, and this allows for the addition of new entities and relationships without disrupting the existing data, which makes them ideal for handling evolving data structures and unpredictable information. Then there's this, efficient representation of complex relationships. And as I said earlier, the knowledge graphs naturally represent relationships between entities using these nodes and edges. So this makes them well suited for capturing the interconnected nature of data in things like social networks or biological systems or supply chains. Next, please. The knowledge graphs also offer this efficient way to do graph traversal algorithms. And this is really good because it allows us to do rapid querying of the connected data, even across multiple hops, which is particularly useful for tasks like, you will see it on recommendation systems when you're in social media, fraud detection, and of course, in our case, for semantic search. Next click. Um, Knowledge graphs are better at handling unstructured data, right? They can incorporate the unstructured data like text, images, videos, and they can represent them as entities and connecting them with other entities. This enables a more comprehensive analysis and insights. Click, please. And they can integrate data from various sources, which is very good for us. They include structured and unstructured data into a unified representation, which helps us with data sharing and analysis and decision-making across different systems. Next, please. And so where we are then is this is what the knowledge graph looks at its core. If it was stripped of all of the data, this is what that schema looks like. And so we have, at the core, this triple of gene and phenotype and disease. And then we have all of the additional information that allows us to integrate all of the knowledge that we know from all of the sources that the Monarch Knowledge Graph integrates. Having a unified data model for it, it also integrates information from ontologies and connects it. So an example, click please, is here represented as as a gene. So for instance, PSEN1 and APOE genes are both known to be associated with, down at the bottom on the right, Alzheimer's disease. And one of the characteristic phenotypes of Alzheimer's disease are the cerebral amyloid angiopathies, which can be represented, as a modifier when they come in as a late onset. And so these little examples here are just a way to give you a guide of where the information goes, kind of like how the information is represented by the knowledge graph. Next, please. Monarch basically then has this job of integrating a very heterogeneous collection of data and ultimately support the diagnosis of diseases and treatment discovery. So as this is just kind of like a graphical representation of what I was sharing, starting with the disparate data, in the beginning there was chaos, and then how we use the semantic layer in order to integrate it through a unified data model to ultimately give us the tools that allow us to do disease diagnosis and treatment. Next, please. Okay, so here I want to, give us a little bit of information about what is the value proposition. So, this is a very powerful knowledge graph. And so first click, the first thing is that it brings together data from diverse sources, right? It includes genetic and phenotypic and clinical information. It provides a holistic view of human biology and disease at its core. And it also has high quality curation, right? It's carefully curated and standardized, ensuring that we have both accuracy and reliability on the data. Next click, please. It also gives us, it has powerful querying. The API enables complex queries. It explores relationship between genes, diseases, phenotypes, but it also allows us to leverage semantic technology. And the knowledge graph has, allows in this way for more nuanced and precise search capabilities. Click, please. By focusing on comparative genomics, it integrates data from model organisms, and so researchers can gain valuable insights into disease mechanisms and potential therapies. And it also has an evolutionary context, in understanding this context of human disease can help us in the development of targeted treatments. Next, please. And it also helps us with faster discovery, right? Accelerating rare disease research, because we're trying to provide a centralized resource that where the knowledge graph can accelerate the pace of the discovery. And it can also help clinicians diagnose, can help them as a tool for diagnosis in more accurately and efficient manners. It also can help with the development of new therapies by identifying the potential drug targets and enabling in that way the repurposing of existing drugs. Next click, please. The knowledge graph also promotes data sharing and promotes collaboration amongst researchers. It also, by using standardized ontologies and data formats, it enables seamless data integration and analysis. And of course, allows we would like to think that because we're leveraging all of this technology, our data is ready for machine learning analysis. So one more, please. And then, for the future, we're, one of the graduate students in our team and many of our technical team people are hoping to give more remixability to the graph. I like the word, I didn't coin it. So in essence, the Monarch knowledge graph empowers researchers, empowers clinicians, and all scientists to unlock the complexities of what we see in human genetics and rare diseases. And this helps us fulfill Monarch's mission on accelerating precision medicine through open data science. Thank you very much. I'm going to let Kevin take it from here.
All right. So I can talk first about how you access the Monarch graph from the web. And this is the Monarch Initiative homepage. And it's an organizational homepage. So it represents not just the sort of the website and the knowledge graph, but the whole big picture team. And up front are the foundational products and HBO, Mondo, Eximizer, Uberon, the knowledge graph, and feeder packets all have direct links. If you follow the knowledge graph circle or go into the search box, you'll arrive at this what we call the Explore page, which gives you sort of some initial search results. You haven't done a search, but it's giving you some suggestions and lets you get to uh, searching the graph, annotating text, Phenotype Explorer, and we will um, touch on those as we go. So this initial destination is Wiedemann-Steiner syndrome, and this is our what we call a node page for it. But this is the disease details, and I wanted to really highlight how what we're doing with the knowledge graph is bringing together all of the great things that are made across Monarch. And so this disease itself is from Mondo, and that's where the description comes from. And of course, it's the place in the hierarchy. Up top, we have the mode of inheritance and causal genes that are coming from the HPO side, and then we have some sort of special links that are built from the Mondo Sesam mappings. Um, and uh, and then sort of like coming down the page, pretending that I'm doing a live demo instead of showing slides, pretend I just scrolled down. Um, we have this bar graph that breaks down phenotypes by system, and then secondarily, we have the phenotype shown as this table, which is sorted by frequency and currently shows sort of associations as like graph edges with details. And so there you can see some of those qualifiers that were on the model graph, and frequency is a very important one. And a special call out for that, everything that's showing at this top of this page is a very high frequency phenotype, but it's always important to know that phenotypes could be either not present or found at very low frequency. And so like just the connection between a disease and a phenotype doesn't tell you enough, and you need those qualifiers for the extra details. But the next place we're going to go is I would call out this Phenotype Explorer button. And this lets us go on a side quest and ditch the node page, and bring our list of 102 phenotypes from Wiedemann-Steiner syndrome over to the Phenotype Explorer to find, in this case, mouse genes that are the most similar to this disease. And so it's going to take this list of phenotypes, and we've chosen mouse genes as the thing we want to search for. It's also an option to compare against other lists, but I didn't make a slide for that. So we do that search, and it brings back on the far left list of matches of mouse genes and the average similarity score across all terms. And as you can see, the little ring icon is not full, and so that's kind of showing us we didn't find an amazing perfect match. But also for context, these mouse genes and this disease have phenotype profiles of 100 terms. And so it's not necessarily going to make perfect matches across so many. But what the Phenotype Explorer and the PhenoGrid below let you do is get a deeper look at how the terms match. This EDNRA gene has, let's see, is that, I'm trying to remember which one is the mouse phenotype and which one is the human, but lowered ear position and low set ears. I believe probably low set ears would be the human term. Lowered ear position is the mouse phenotype term. And so these are very similar terms from two different ontologies. And this ancestor section is showing you that they took what is probably one step up to a very specific term. That's why that ancestor IC score is 16, that's the same scale as these scores on the left are averaging. So 16 is like a really like good score. That means that they met at a very specific term, which was decreased external ear position. And that's not a term from the human phenotype ontology or from the mouse phenotype ontology. That's a eupheno term that is generalizing across organisms. And so that's where they're meeting. And then we have a similar situation that is not as specific for decreased body size and short stature lining up, and which is decreased size of the multicellular organism. And so this is sort of uh, coming in and where they're coming together. And then so I'll follow, wasn't a great match, but I'll still follow that mouse gene. And that'll give us a chance to look at what else shows up on a gene page in the Monarch UI. Um, we have the same overview for the genes, and then we get the histopheno bar chart again. And to call it again, this is something that we're getting thanks to the Eupheno team. Um, these high level terms, growth, skeletal system, digestive system, are from the Eupheno ontology, and they are cross species. And so this graph is always going out and asking for children of those eupheno terms on any gene or disease page that you land on, and will show you sort of grouped by system how many phenotypes there are. And then additionally, there are a whole bunch more tables. There's gene phenotype associations, interactions, pathway, expression, orthology, and the 3 Go tables. This gene may have only had 2, or I may have not pasted my last screenshot, but there are all these data tables available. And then there's an interesting feature below that called breadcrumbs, and this is a different traversal. So here just to explain what it does, it is capturing your path as you move through the website. And so essentially, it's your browser history. But what you don't get in a browser history is the associations between each page that you viewed. And so when you start at Achu syndrome, which I am still astonished that that is a real disease, and that is the effect of sneezing when you see the sun. And it is apparently heritable, and so it's an OMIM, and so it's in Mondo. It has the phenotype of paroxysmal sneezing, and you can wander up to sneeze, and from sneeze, you can wander up the ontology to constitutional system, and then you can go to a phenotype, and then I believe I ended up on a different disease from there. And so like I was on this familial cold autoinflammatory syndrome 1 page, and at the bottom of that page, there is the sort of section describing how I got there from Achu syndrome. And then below that, we've got the hierarchy. So this is Ehlers-Danlos syndrome, showing, that it is a child of syndromic disease, hereditary disease, and developmental defect during embryogenesis. So those go up the ontology, and then going down the ontology, we have all of the subtypes of Ehlers-Danlos displayed. And we have this hierarchy section for every term from the ontology. And then coming to the end of the UI, we have the text annotator. And what this does is apply some natural language processing to find any phenotype terms, diseases, genes, Go terms, anything that's in text. It gets sort of extracted as an entity and then checked against entities in the Monarch knowledge graph. And then sorry, I looked at the chat.
That's our bad. Sorry. We're still talking about schema. Sorry.
No, I want to talk about schema. And so it pulls out entities from the text, checks them to see if they're an entity that's in the Monarch knowledge graph, and it gives you can hover over them and see if several entities match. Like in this example, that I don't want to try to pronounce what I get for picking that example, but you can see that it is not 100% text match. It is sort of a partial match and maybe a synonym match against those. Um, but again, we have a phenotype explorer button, so you can take the list of phenotype terms that were found in a piece of text and bring them over to Phenotype Explorer and search for the diseases that are our strongest match. And in this case, you can see those 14.5 scores are about a much better, much stronger average score than we had looking at mass genes. So next is the Monarch API, so sort of stepping down in the stack, I guess. And this allows you to retrieve associations, do searches, do the text annotation. It essentially, like everything that drives the website is present here. And at this point, it is not, the API is not necessarily engineered for like general data access. You can get to everything, but it's been designed as with the website as the primary consumer, I would say. Other than there is, I guess I can have a call out to halfway down through the right-hand side, there's a multi-compare feature, and that lets you put in several sets of phenotypes and get the equivalent data structure of a phenogrid for those phenotypes. And that's sort of designed especially for friends on PC. So we also have the Phenomics Assistant. And so this is not integrated into the primary website yet, but this is an AI chatbot that uses the Monarch API to sort of gather data and lets you sort of ask a question like, what is the genetic basis of cystic fibrosis? And it will sort of go out and fetch what are the causal genes and make the query and sort of answer your questions. And at some point, we may build that into the site to make it more available. Section is near and dear to me, which is accessing the graph as files, because that's much of the time as a person building the graph is it's a bunch of files. So there's three sort of levels or at which you can sort of get data from Monarch knowledge graph. The first is, and I'm going to use a pizza analogy for them. The first is the modular KG components, and these are ingredients. This is your double-aught pizza flour, your can of tomatoes, your beautiful mozzarella, and when you're using these, you're getting them sort of like the least processed files, and so they are brought into the same schema. So they're brought into BioLink. That means you're going to find coherent columns across all of them. If you want gene to phenotype data from a whole bunch of different organisms, and you want them all to be in the same format, like this will be there. And it's our sort of translation into the BioLink model. And I guess I shouldn't go into too much detail. I think I have slides for each of these. Anyway, the KG, which is the complete normalized graph, and this is kind of like our reference structure. And the reason to use this is probably that you want a whole working knowledge graph, plus or minus a little bit. You basically want the whole thing, and you're going to adjust it from there. And then finally, the TSV exports are a little bit more like treating the Monarch knowledge graph as like a regular knowledge-based site, like Lines of Genome Resources or any other, where you go and there's a downloads page and you sort of want TSVs, and those TSVs that are grouped really by subject. And so you can get the gene phenotype across species, et cetera. So the KG component example here is NCBI gene, and this was a real motivator of our push towards making these sort of modular individual transforms. And that was because the Mondo team and the Monarch team both needed to have a representation of NCBI genes. And this process is a subset of species, and it's in Git, it runs as its own thing, it's got each file, and you can use those as pieces. So if you are building your own data project from scratch and you want NCBI genes from like a certain subset of species, this is where you can get them already converted to biolink format. And if the taxa you need isn't there, you can do a pull request or an issue, and we can add it very easily. The KG as TSV files, that's like you're going to load it into KGX to put it in Neo4j, or you're going to use Grape to do some kind of graph ML, and you want the sort of whole KG. And then like TSV exports is like very similar to the input file, but what's different is it's post normalization. And so if there was an upstream entity that a gene referred to that wasn't present in our genes, it might not make it. It's after the merge and then split apart, and it is broken up category. So if you want one file that has all the gene phenotype, that gene phenotype all.tsv is a place to get it. And then sort of wrapping up the available important files is our test mapping collection from Monarch Mapping Commons. And this is what allows entity normalization in the graph. And we sort of produce it and share it, and it's a first class data product that you can use to convert your DO IDs to Mondo, or your NCBI genes to fly-based gene, etc. Okay, finally, accessing the graph as a graph. Daniel's gonna take over with this part.
Yeah, next slide. So Kevin kind of hinted at this in the last section about working with the graph as files. Essentially, there is a very useful software system for hosting graph databases called Neo4j. Why would we want to use this over interacting with files? Basically, we put the graph database up like this, and if you have questions you want to know about what information is in the graph, all you have to do is know the right URL, the right address, and you say, hey, what do you know about stiff person syndrome? Hey, what do you know about this gene? And we'll give you everything that's currently in the graph as a file. So it uses this query language called Cypher. You can read the guide if you go to the slides, and but essentially it's very similar to how SQL works, and it essentially is tailored to work with graph data. The main benefits of using the Neo4j system over directly kind of trying to edit the TSVs is that there are already solid drivers for making these queries to the database in like R and Python. Very nice visualization. We'll see that in a second. And other knowledge graph teams, specifically the Renzi team and the Hedionet team and this other team that makes this graph called ARAX, all have Neo4j endpoints. So if you write a query pipeline for our graph and you're like, well, this works well here, doesn't work well if we use it on another graph, you just change the URL at the top of your Python script or whatever, and you immediately will get new results. A little bit of tweaking here and there, but that's generally how it will work. Oh, okay, next slide. So I'm just going to go through really quick how just a really straightforward, like how I would go about trying to do some Neo4j querying. The address in the top right is the way you can access this web portal here. So all I did is I just told the graph, send me a single node. So match n, return n, that's it. You see the graph, send it back to me. If you go back and want to get more information, those little black arrows, just click those and you'll kind of start seeing the information that the actual graph is sending back to the browser. Next slide. So here's a little bit more complicated of a query. Now, instead of just asking for a node, we ask it to send us two nodes that are connected by some form of relationship. So we see here that pi1 has it interacts with edge with SPN4. So we can start seeing how we can we start very small with one node, we can build out a little bit. Next slide. So now we ask a question like what do we know about diseases? So here we just say, give us a node of type disease. You see we get back something. So that's we can start to narrow in on what specifically the graph is going to tell us about. Next slide. So this is really useful for most queries. If like I would say if you want to do this, come back and look at these slides or watch this video later, but this will let you say essentially search the graph. So what this query says here is any name of a node that has the word cystic fibrosis, give that back to me so we can look through them, because you won't have 100% knowledge of what information's in the graph when you're asking a question. So being able to just quickly say, oh, what do we know about autism? What do we know about Alzheimer's? And get every single node that you can like look through. So, oh, we have these 6 variants. Oh, we have this. That helps really quickly start to ask better questions. So next slide. So now I've used the knowledge that I gained from the last query that cystic fibrosis is in fact a node in the edge or a node in the graph. And now I'm just saying, hey, find cystic fibrosis and then find any genes connected to the cystic fibrosis node and send them back to me. So that's cool. Now we know what genes have been cataloged by the graph, by our team so far. Next slide. So similarly, we can do this for phenotypic features, which enables us, we can see this information set that we have. Next slide. Okay, so now we start to get a little bit more complex, and you can see very quickly you can start building out really interesting, complex series of questions you might want from our data using this query language. So here we go from cystic fibrosis to a phenotype to another disease. So now we can say what diseases have phenotypic similarity to cystic fibrosis. So that's really useful. And that's something that lets you kind of start to ask questions about not only one set of relationships, but multiple sets. Next slide. So now adding one more level onto what we just asked. So we start at cystic fibrosis, we go to a phenotype, we go to a disease, and then we ask what genes are connected to those diseases. So what are the genetic components of phenotypically similar diseases to cystic fibrosis? Really cool. Next slide. So now this is sort of what it would actually be really useful to us, and this, you can quickly build out a query like this. Now we say, okay, do what we did before. So start at cystic fibrosis, go to a phenotype, go to another disease, go to a gene, but only tell us about that gene if that gene's related to cystic fibrosis. So we've essentially said, tell us all diseases that have genetically and phenotypic similarities to cystic fibrosis with this query. So that might be that's a pretty typical use case, is to like sort of find one or two ways that another disease or another like disorder is connected to a single entity, and you can do that pretty quick with Neo4j. Next slide. Okay, so this is like a really I'd say like an advanced query, but essentially I asked, can you give me all of the genes that have and tell me all of the diseases that are phenotypically similar to cystic fibrosis that share that gene. So we'll see the results of this query in the next slide. So now we can see that CFTR has 5 diseases in common with cystic fibrosis. So yeah, you can, this is really powerful. This does not take very long if you get good at it. And all these queries ran in under like 10 seconds. So yeah, super useful. Thank you.
All right. I love that. Thank you. Up next, we have an R library now, and it's somewhat experimental, but it allows you, it's a different approach from our API. It talks directly to Neo4j and is built from the idea that you're going to have a graph representation locally in R, but it's not going to be the whole graph. You're going to sort of get a collection of nodes and edges, and then you can sort of grow them out. And so in the same way that Daniel walked through that query, sort of starting at a gene and walking out through certain predicates and properties, then this kind of has that approach. And then I think that it has an interesting extra addition, and that it can also query our semantic similarity endpoint. And so you can walk through not just diseases that share the same phenotype, but you can sort of have that algorithmic connection between the two genes where, it's about sharing an average, an average high scoring connection through a particular graph traversal. And so that one would be really challenging to represent as a Neo4j query. But in this context, you get to kind of blend a pure graph querying with the sort of great stuff that the algorithms team has done. And I think it's looking for more users to sort of drive use cases, but it is sort of at least beta usable. And that wraps up, and we have a team slide.
Yes, thank you so much. Kevin and I were just wanting to make sure that we recognized all of the work that so many of you contribute to the development of the knowledge graph and all of its services. On the far left, we have our core development team. In the middle, many of the contributors. If you are supposed to be here and I didn't write your name, we sincerely apologize. Please give me that so we can add it to the slide. And then on the right, we have the principal investigators for the Monarch initiative. Next slide. And in here, there are some ways to contribute. You have a link that Sarah dropped on the chat for this information. We are also funded through two very different mechanisms at NIH. The Office of the Director has been funding the work of the Monarch Initiative for the past 13 years, not all of them continuously, but it's been an incredible support, or it has been very big proponent and supporter of everything that we do at the Monarch Initiative. So we're incredibly grateful to the Office of the Director, and the National Human Genome Research Institute funds our work on the Phenomics First resource, which of course generates all of the information for UPheno, or funds all of the work for UPheno and Mondo, Phenobuckets, in our integrated platforms. And so we just wanted to make sure that we remarked on the continued support from our funding partners. All right. Well, it has been our absolute pleasure to prepare this for you and to share a little bit about the resources for the Knowledge Graph and all the services. As you can see on the slides, there are ways where you can get involved and contribute. If you have more questions, or if you find if you work for a resource that you think should be included for consideration to be included in the Monarch Knowledge Graph, please let us know. Reach out through a ticket or reach out via email. You can also subscribe to our Friends of Monarch Google group. It's very low activity and it's mostly about updates. Our fearless, amazing Sarah keeps us all updated with information on LinkedIn. So you can follow us on LinkedIn as well. And there's a collection of YouTube videos with this and many other presentations. So make sure that you keep track of everything that we do and visit us at monarchinitiative.org.